Hi everyone, and welcome to episode 29 of the Cancer Researcher podcast brought to you by the European Association for Cancer Research. In this episode, we invited Professor Xin Lu, director of the Ludwig Institute for Cancer Research at the University of Oxford, cancer theme co-lead for NIHR Oxford Biomedical Research Centre, and director of the Oxford Centre for Early Cancer Detection. She will discuss today with us about her research interests throughout her career, starting from her PhD and up to this date, and will give us some teasers about the keynote lecture at the upcoming EACR Congress in June 2026 in Budapest. I am your host, Dr. Alexandra Boitor, Scientific Officer at the EACR, and I invite you to dive into today's topic. Hello, Xin, and thank you so much for joining us on the Cancer Researcher podcast.
Hello, I'm really delighted to have the opportunity to join you for the Cancer Researcher podcast.
The pleasure on our side as well. So you are very well known for your long-standing interest in mechanisms of tumor suppression and cellular plasticity, centered on studies of p53, you being one of the very first researchers to study this tumor suppressor. And then also the ASPP family of proteins, or apoptosis-stimulating protein of p53, which you discovered. Over the years, you looked at different aspects of cancer biology, from tumor suppression to cell death, differentiation, cell growth, gene expression. But what I'm wondering is, how did you find and define your research interest? What drew you into cancer research in the first place, and specifically to cellular plasticity?
Okay, yes, I probably should tell you a little bit about my background, and then that would also naturally lead to how did I get into tumor suppression as my major interest and research topic, which until today I'm still fascinated and interested in this area of research. I started my education in China, and my undergraduate study is in biochemistry. And then at that time, I was fascinated by the emerging research area of molecular oncology. I joined a group in Cancer Institute, Chinese Academy of Medical Sciences, to do my master degree. And it was the beginning of the oncogene era, and where people discovered the human oncogene, like the rat's oncogene. Then subsequently, the tumor suppressor gene concept was also revisited, and people started looking into tumor suppressor genes. So it was the area molecular oncology was emerging, and I was really fascinated by the ability to study cancer at the molecular level. So I did my master degree in Beijing in the Cancer Institute, Chinese Academy of Medical Sciences, and working on oncogene. But of course, I was also started to be interested in tumor suppressor genes as well. I subsequently got a research fellowship from WHO and the International Agency for Research on Cancer, which allowed me to come to the UK at the Imperial Cancer Research Fund for 1 year training. And I studied in a group which also specializes in oncogene cooperation. That's Hartmut Lange's group in Lincoln's Inn Field. After that, I did a detour, and I worked in the lab, which is specializing in intermediate filaments, keratins. And that gave me the basic training of epithelium biology. But then I realized my heart is at the oncogenes and tumor suppressors, and particularly in tumor suppressors. And that was the time when I then joined David Lane's lab and studied p53. Just to give a background, I did my PhD in Birgit Lane's group, and Birgit Lane is the wife of David Lane. So it was very convenient for me to shift lane from Birgit Lane to David Lane. And then I started my postdoc on p53. When I started with David, it was the time we actually don't know how the wild type p53 would act in tumor setting, because it was the time we realized p53 is a tumor suppressor, and therefore in cancers, you're supposed to see p53 is all mutated. But then the cell lines I started to work with they're all containing wild-type p53. And I actually was doing all this p53 sequencing at the time where sequencing a PCR products was difficult. But I managed to sequence them all. They were a wild-type p53. And David Lane would say, oh, you haven't sequenced them all, so it must be mutated. And it's mutated somewhere else. So I sequenced every single p53 sequence in all 9 different cell lines, and they were transformed by RAS and MET oncogene. They're all wild-type p53. But then the 4 cell lines has a RAS mutation. They all have a mutant p53. So there was a pattern emerging where wild-type p53 somehow is maintained in the RAS and MET oncogene transformed mouse prostate cancer cell lines. It's mutated in the RAS-only transformed cell line. So my fascination of looking into what this wild-type p53 is doing in these cancer cell lines and led me to make 2 observations during my postdoc at the time. One, p53 staining was all clustered in the micronuclei. Those are the nuclei, much smaller than the normal. And then they stain very strongly. Doing my PhD in Clairvaux laboratories, and that's where all the DNA damage and repair activities were, educated me about what micronuclei means. That means there's something with the damaged DNA in these micronuclei. So that gave me an idea. Maybe p53 can be accumulated in DNA damaged cells. The second observation I saw with these RAS-MET transformed cells, they change the color of the medium really rapidly and they die a lot. And so that also suggested when they have the wild-type p53 in these RAS-MET transformed cells, somehow they cell die much more than the ones with the mutant RAS transformed cell. That was the beginning of my postdoc. And basically I spent the next 3 years follow all of these. And it turns out p53 is a major sensor to damage the DNA. And I thought I have the whole time on my side to work out exactly what triggers p53 response. And of course, there was a paper published where I was still trying to figure it out and precisely what DNA lesion was causing the DNA damage response. So that led us as one of the early groups to report the p53 response to DNA damage. And then also during that time, we also observed p53 response to UV and gamma radiation has a different kinetics and also different response. Until today, I still don't think we completely worked it out why there's such a difference in the kinetics in p53 response to UV versus gamma radiation. But it certainly attracts my interest in working on a single molecule, which turns out to be the most important tumor suppressor in human cancer.
It's always fascinating to hear research stories of established researchers and see how nicely things fit into the puzzle. In the end, I'm sure it wasn't so linear throughout, but it's so nice to see how nicely it aligns and how much one can achieve within their career. But I also find it fascinating as a reminder of how new the research field actually is. And as you said at the beginning of your career, it was just a forming research field, molecular biology of cancer. And now some of the things that you've helped discover and you studied have basically become dogma. So it's always fascinating to see that.
The second part I might want to tell you then is my own view of cell plasticity and why I still work on p53 related area of research, and then how did that lead us to discover the ASPP family of proteins. As I mentioned, I was really fascinated about how p53 responds to DNA damage and oncogene stress. And when I started my own group, and it was obviously very important in my mind, I need to look for something which is my own, but also most importantly, which could play a major role in regulating the tumor suppressive function of p53. The time when I started p53 was the molecule of the year. And what we know then P53 is a transcription factor. It could turn on and off lots of genes. But the most well-known target of P53 at the time was the inducing cell cycle arrest. Also, some of the target has been identified. It might induce apoptosis, which is a type of cell death. My idea was, well, cell cycle arrest is not permanent. And if you truly want to damage the cells, you really want to kill the cells and completely eliminate it. And the question is, can we find a way selectively pushing P53 towards inducing cell death? And in a way, whatever is selectively regulating P53 ability to inducing death would be far more important than it only causing cell cycle arrest. And that was the thinking behind it. So I started looking for things which can selectively pushing P53 towards inducing cell death when the cells are damaged. And that's really how we discovered the ASP family of proteins. ASP stands for apoptosis stimulating protein of P53, but it is also a group of protein containing protein sequences called ankyrin repeats, SH3 domain, and proline-rich sequence containing proteins. And this is a family of proteins. It's evolutionarily highly conserved in C. elegans and Drosophila, which is the invertebrate. There's only one member of the ASP family. There's also only one member of P53 in invertebrates. But by the time to vertebrate, there are three members of the ASPs, which has ASP1, ASP2. They both are the activators of P53-induced death. And then there's an inhibitory form of ASP called IASP, which inhibits P53-induced apoptosis. It's the same also by the time it gets to vertebrate. There are three members of P53 family. They are P53, 63, and 73. So that coincidence of the evolutionary development between the ASP and P53 family is certainly still interesting and fascinating. And that's where we are at the moment.
And the ASP family of proteins brings incredible promise to cancer prevention, detection, and treatment. ASP proteins could serve as novel biomarkers and excellent targets for the development of cancer therapies. So it's always really interesting to hear more about the journey of discovering those proteins. Continuing on this note of research projects that you've developed and worked on, I wanted to ask you, what is your favorite research project, the one that you think was the most interesting that you've ever worked on? And it can be something that you're currently working on in your group or something you might have worked on in the past.
Well, I'm obviously biased, and so I think the most interesting remains to be how P53 is regulated, and I think the ASPs are very interesting and fascinating regulators of P53. So regulation of P53 and the whole role of the regulating P53 function in tumor suppression remains to be one of my most interesting projects. But also that would lead me to why we are interested in cell plasticity and why this regulation is fascinating. So when we talk about cell plasticity, I would think this is perhaps the most important biological process that's very much involved in cancer initiation, cancer progression, and cancer resistance to therapy. Why? Well, cell plasticity, in my own interpretation, is the ability of cells to change in response to external signal. And what would be the most important things to prevent cells to change the plasticity? This is the key. And what are the molecular switches which turns on and off cell plasticity? And I think this is the fundamental area of cancer research. If we truly understand how the cell plasticity is controlled, can be turned on and off in response to defined signal, we will make major advancement in understanding of cancer initiation, progression, and resistance to therapy. The best example of cell plasticity is probably induced pluripotency cells, IPS. If you go back to the history, you only need 4 transcription factors to induce any of the differentiated cells, like the fibroblast cells, and reprogram it all the way to the pluripotent stem cells. And that process is not actually very effective. However, you could enhance the IPS efficiency by knocking out tumor suppressors, and P53 is the number one. If you knock out P53 and give in the 4 transcription factors, you can enhance the reprogramming efficiency by 1,000-fold. If you knock out many other tumor suppressors, you could do the same. May not be the same extent of the efficiency enhancement. What this is really telling us is tumor suppressors are the major brake for allowing cell to change cell fate. It controls the ability of the cells to turn on the cell plasticity. MYC oncogene is one of the 4 transcription factors which promote the reprogramming. So you can see how intimately it's linked between reprogramming the cell plasticity change and the cancer initiation and suppression. The second part, which is also very important about cell plasticity change, is in response to infection. So many of these infectious agents are very potent inducer for cell plasticity change. And one of the things my lab has been fascinated by recently, well, in the last 10 years or so actually, is about the H. pylori. This is the very well-known bacteria. It's infecting about half of the world population. Unfortunately, only very few people get gastritis and then also get gastric cancer. But there are 2 major types of H. pylori. One contains an oncoprotein called CAG-A, and the other are not. So most of the Asian strains, the ones present in Southeast Asia and China, contain CAG-A, and most of the H. pylori strain in the West do not contain CAG-A. So if the H. pylori containing CAG-A, and that bacteria will have up to 6-fold enhanced ability to induce gastric cancer. And one of the major biological functions of CAG-A is to induce cell plasticity change. So if epithelium cells has CAG-A, the chance of changing from the gastric epithelium to mesenchymal cells, a process called EMT, is dramatically increased. And it turns out the most important cellular targets of CAG-A is the protein we discovered, ASPP2. Not only we know the CAG-A binds ASP2, we also collaborated with a group and solved co-crystal structure between CAG-A and ASP2. And there is a paper coming out in Cancer Discovery by studying the H. pylori CAG-A in 6 different countries and looking for the link with cancer incidence, the virulence of CAG-A. And the determining factor is its ability to bind to ASP2. The more CAG-A in the bacterial strain to be able to bind ASP2, and the more oncogenic that H. pylori strain is. And we do have evidence how the ASP2 CAG-A complex is able to controlling the cell plasticity change. So that is one of the area, the angle, how we see the cell plasticity involves in cancer initiation and progression, and why we are fascinated and interested in studying cell plasticity in gastric cancer. And then the final one is gastric cancer, and it's not the only place where cell plasticity change happens. And in the upper GI, where the esophageal cancer, particularly esophageal adenocarcinoma and Barrett esophagus, is also very well recognized and accepted as a place where cell plasticity is changed quite dramatically. And this is the reason why we are very interested in focusing on studying the role of cell plasticity, the regulation of the ASP and P53, and in the upper GI cancer, and particularly in response to infection like H. pylori.
This also very nicely answers one of my other questions, which was regarding how or why did you switch the cancer type that you work with in your career from melanoma at the beginning to esophageal and gastric cancers now. So thank you for that. Is this something that we might find out more about this story at your lecture at the EACR Congress, about how these H. pylori-induced gastric cancers develop?
Yes, I would definitely plan to talk about the H. pylori and an ASP2 link in regulating cell plasticity. And I will also talk about our recent work on immunochemotherapy of esophageal adenocarcinoma. And that certainly is one of the areas we are talking about at the conference.
Well, thank you very much. I'm really looking forward to learning more about this. And thank you so much for telling us more about the connection between P53, the ASP proteins, and MYC. I think our conversation has been almost like a fascinating lecture, to be honest. As we're approaching the end of the episode, I wanted to ask you if you could tell me one thing that you're looking forward to at the EACR Congress in June.
Yes, I'm looking forward to meet old friends and new friends particularly, and then to learn the new technology and latest development in cancer research. And of course, most exciting area I'm hoping for is establishing new collaborations with all the people there. That's what I'm really looking for, and that's what the conference is about. Allow people to learn things and to meet new people and set up new collaborations.
I think conferences in general and the ACR Congress in particular are great both for learning the latest developments, but also to network with old friends and to develop new collaborations. This has been brilliant. You as a person and your career are truly inspirational. I'd like to ask you so many more questions, but unfortunately, we're just about out of time. So we'll leave it there for now. Once again, thank you so much for joining me on the Cancer Researcher podcast.
Thank you for inviting me, and I'm looking forward to the EACR meeting.
We really hope that you enjoyed listening to this conversation with Professor Xin Lu, and that we got you interested in joining us for the annual EACR Congress to take place in Budapest from the 8th to the 11th of June 2026. Please subscribe to our podcast so you won't miss an episode. You can find The Cancer Researcher on your podcast platform of choice. Subscribe and tell your friends about it. And don't forget that you can also find us on Blue Sky and Instagram at Hello EACR, and you can search for us on Facebook and LinkedIn. We welcome feedback and suggestions for other topics or preferred guests through our feedback form, which you can find on our website at www.eacr.org/podcast. Until next time.